Welcome my dear students. Today we explore Mendelism, the foundation of genetics. Gregor Johann Mendel's simple pea-plant experiments revealed how characters or traits are passed from parents to offspring through definite rules. In this class we will uncover his fundamental laws of inheritance in a clear and exam-friendly way. Let's begin our journey into the principles of heredity that shape every living organism. Genetics and Gregor Johann Mendel. Genetics is basically the study of heredity, that is transfer of traits from parents to offspring. The term genetics was proposed by William Bettison in 1905 to describe the scientific study of heredity and variation. So, at the time of Gregor Johann Mendel, there was no term as genetics, and some students thought that the genetics term was originated from the term gene, but that's not the thing. William Johansen introduced the term gene for Mendel's factors in 1909. So what we found here, that the word gene comes after the term genetics was proposed. Genetics was proposed in 1905 and the gene was proposed in 1909. So the word genetics is a derivative of the word gene. The Gregor Johann Mendel laid the foundation for the first time of modern genetics before the chromosomes and genes were discovered. He used simple pre-planned experiments analyzed quantitatively with large sample size. From his work, fundamental principles of heredity were established. We now know genes are located on chromosomes and can be replicated, expressed or mutated. His laws form the basis of classical or Mendelian genetics. Not all genes show Mendelian inheritance, but Mendel's work remains the starting point of inheritance studies. So Gregor Johann Mendel is considered to be the father of genetics. Historical steps in the foundation of genetics: pangenesis. Hippocrates in 5th to 4th century BC firstly proposed about pangenesis. The idea is all body parts produce tiny particles called pangens, and from the pangen the theory of pangenesis was derived. According to this theory, the particles collect in the reproductive organs and they are transmitted to the offspring. Suggested offspring resemble a blend of parental traits since all body parts contribute them. After the theory of pangenesis, pre-formation theory was established in 17th or 18th century. The belief was that organisms develop from a fully formed miniature individual, that is known as homunculus, which is present in sperm or egg. The implied traits are pre-shared and inherited from one parent, supporting the notion that offspring traits could combine or blend. Germplasm theory. August Weismann in 19th century proposed about the germplasm theory. According to his concept, the germ cells, whether it is egg or sperm, carry hereditary information and somatic cells do not affect the inheritance. While emphasizing discrete units, it was initially interpreted as hereditary material blending when male and female gametes combined. Epigenesis theory. Aristotle was the profounder of this concept in 17th century. Idea is organisms develop gradually from undifferentiated material rather than being preformed. So it suggested that traits from both parents contribute to development, implying intermediate or blended offspring traits. Now we should study blending inheritance, which is the old theory. The concept was proposed before Mendel in 19th century by early biologists like Joseph Kalreuter. He suggested that parental traits mix like paints to form an intermediate trait in offspring, so such as red and white paint mix to form the color pink. So when tall father is crossed with short mother, medium height child will be formed. So the key points of the hereditary factors blend permanently, and this theory couldn't explain reappearance of traits after skipping generations, dominant recessive relationship and their maintenance of variation in nature. The limitation, it couldn't explain the trait reappearance or variation. From blending inheritance to Mendelian particulate inheritance. 19th century view, it was the blending inheritance. Scientists thought offspring traits were a blend of parental traits. Example, tall into a short, tall or plant, and when crossed with the short plant, medium height plant was developed. The problem is variation would disappear over generations and traits couldn't reappear. Mendel's observations. Mendel proposed that mammals studied pea plants with distinct and contrasting traits. The discontinuous variation he studied about. He crossed true breeding plants and observed F1 and F2 generations. Found that traits do not blend but remain discrete units. F1 is the only dominant traits appear, but in case of F2, recessive trait reappear in 3:1 ratio. Concept of particles, from where the particulate inheritance theory has been developed. So the particles are either factors or genes. So the traits are controlled by pairs of factors which are considered as alleles. After some days, each parent contributes one factor and they are preserved in the offspring. Traits remain intact and they are not mixed or lost due to time. Gregor Johann Mendel was a teacher, scientist and monk at the Augustinian Abbey in Czech Republic. He taught physics, botany and natural science with monastery support in a church. He began inheritance studies in 1856. He worked first on honeybees, then focused on pea plants. He conducted 30,000 roughly pea plants experiments over 10 years, and in 1865 he presented findings. Traits are passed faithfully from parents to offspring. Traits inherit independently and show dominant or recessive patterns. He published research in 1866, experiments in plant hybridization. His findings were ignored initially due to belief in blending inheritance. Mendel studied discontinuous traits like violet versus white flowers, which showed clear inheritance pattern. He became abbot in 1868, limiting his research work, but further the concept was rediscovered in 19th century, leading to the recognition as father of genetics. Now we should study Mendel's model system. So why Mendel chose garden pea or Pisum sativum? It is naturally self-fertilizing, so it reduces the chance of cross-pollination. Flowers remain closed. They ensure true breeding lines or the offspring resemble parents. True breeding plants prevent unexpected trait variation. So their short life cycle, that is multiple generations in a short time. They are easy to grow in large numbers and reliable statistical results. It shows clear contrasting traits such as tall versus dwarf. So these are the characters, that is seeds, form of the seeds. It can be roundish or wrinkled. The cotyledons can be yellow or green. Flower color can be white or violet red. Pod can be full or constricted or yellow or green. Stem can be axial or terminal, and the size of the stem can be long or short. Mendelian experiment and crosses. Mendel performed hybridization, crossing two true breeding plants with different traits. He manually transferred pollen from one plant to another for controlled crosses. To prevent self-fertilization, he removed anthers before pollen matured. So the P generation is the original parental plants used in the first cross. Seeds from the P generation produce the F1 or first filial generation, and the F1 plants were then self-fertilized to produce F2 generation. Further, Mendel studied traits ratios in P to F1 and further subsequently into F2, which lead to his laws of inheritance. He continued with later generations F3, F4, but P, F1 and F2 results were more crucial. Garden pea characteristics and Mendel's findings. Mendel studied 7 traits in pea plants, each with 2 contrasting forms. Examples: tall versus short, wrinkled versus round seeds, green versus yellow seeds, violet versus white flowers, etc. So trait is the variation in the physical appearance of a heritable characteristics. He conducted large-scale experiment. Mendel analyzed nearly 9,959 F2 plants. The results of Mendel's garden P hybridization, the image showing the results of Mendel's garden P hybridization. So the example, true breeding conformation, white when white flower is when crossed with white flower, all white color flower is found. When violet flower is crossed with another violet flower, all violet flower is produced. F1 generation, all F1 plants had violet flowers, and when he contradicted blending theory, no pale and violet mix. F2 generation, F1 self-crossed, 705 violet, 2 to 4 white, and approximately 3:1 ratio. Reciprocal crosses ratio remained the same regardless of which parent contributed the trait. Other traits, same pattern observed in all 7 traits. One trait disappears in F1 and reappears in F2, that is at 3:1 ratio. Particulate theory of inheritance or Mendel's concept. Traits are controlled by discrete particles. Later it was called as genes. These particles do not blend, they remain intact. Key features, each organism has 2 alleles for each gene. Alleles may be homozygous, same, or heterozygous, different. Alleles segregate during gamete formation or law of segregation. Alleles for different genes assort independently, that is law of independent assortment. So why it was revolutionary? It explained the reappearance of traits after skipping generations. It explained how variation is maintained in nature. It introduced genotype and phenotype concepts. Pair factor rule or Mendel. What it says, each trait is controlled by a pair of factors or allele. One allele is obtained from each parent. Why 2 alleles? Because organisms are diploid, that is 2 sets of chromosome, so 2 copies of each gene is there. Capital T when crossed with small t, all F1 organisms are found. So capital T small t is found in the F1 organism, and all of them are tall, so that each parent contributes one factor. Importance, foundation of genotype, phenotype, dominant, recessive concepts. It explains why recessive traits can remain hidden, that is in case of heterozygotes. That is the image showing the 7 discrete characters and their dominant trait and the recessive traits of the 7 characters. Mendel's law of dominance statement, in a heterozygote capital T small t, one allele is dominant and the other is recessive. Dominant allele masks the recessive one. Evidence, when capital T is crossed with small t, all tall capital T small t is found, and F2 3 tall 1 dwarf. Exception is the incomplete dominance, codominance, over-dominance, and multiple alleles interacting variable. Mendel's law of segregation, purity of gametes statement, the 2 alleles of a gene separate during gamete formation. Each gamete gets only one allele, pure and unchanged. Biological basis, meiosis 1 causes homologous chromosomes separate and alleles also get segregated. Consequences, it explains reappearance of recessive traits which produces 1:2:1 genotypic ratio in the F2 generation. So what is law of segregation? Law of segregation states that the 2 copies of each genetic factor segregate during the development of gametes to ensure that each parent's offspring attains one factor. Mendel's law of independent assortment, it is related to Mendel's law of independent assortment, and it happens during metaphase 1 of meiosis. What happens? When homologous chromosomes pairs align at the equator randomly, the way on one chromosome pair orients has no influence on the orientation of another pair. Therefore, alleles of different genes enter gametes independently. The key idea is the assortment of alleles, that is random assortment of chromosome pairs. Example, gene for seed shape, that is capital R small r, gene for seed color, that is capital Y small y. Orientation can produce gametes like capital R capital Y, capital R small r capital Y, and small r small y. All combinations are possible. This results in genetic variation. 9 is to 3 is to 3 is to phenotypic ratio in dihybrid cross in F2 generation, and recombination of traits never seen before. Chromosomal theory of inheritance. This concept was proposed by Walter Sutton and Theodore Boveri. The main idea is the genes are located on chromosomes, and chromosomes' behavior during meiosis can explain Mendel's law. So the chromosomes occur in pairs and they segregate during the process of meiosis. And fertilization restores diploid condition. Evidence: chromosome movement in meiosis, Morgan's experiment on sex-linked inheritance or Drosophila. Chromosomal basis of segregation, it is related to Mendel's law of segregation, and it occurs during meiosis 1 or anaphase 1. What happens? Homologous chromosomes carry one allele each for a gene. During anaphase 1, these homologous chromosome pairs separate and move to opposite poles. As a result, each gamete receives only one allele of the gene, which is pure and unchanged. The key idea is the segregation of alleles, that is segregation of homologous chromosome. So what is the example? Capital T, tall, indicates tall, and small t is dwarf. A heterozygous plant, capital T, small t, produces two types of gametes, one with capital T and one with small t. No mixing occurs. Alleles remain separate. These explain the purity of gametes. Reappearance of recessive traits in F2, and 1 is to 2 is to 1 genotypic ratio in monohybrid cross. So, to conclude today, Mendelism gives us the basic blueprint of how traits are inherited across generations. By understanding Mendel's law, you can easily decode genetic crosses and predict outcomes with confidence. Keep practicing, because mastering these principles make genetics simple and enjoyable. See you in the next lesson. Thank you.